This video describes the proline biosynthesis pathway project from the winter of 2014 to 2015. What is proline? Proline is one of the 20 common amino acids. The distinctive cyclic structure of proline side chain gives it an exceptional conformational rigidity compared to other amino acids. Because it increases a protein's conformation stability, this may account for proline's higher prevalence in proteins of thermophilic organisms. Proline disrupts alpha and beta helices and secondary protein structures because of its cyclical shape. It's a free radical scavenger. And finally, proline is an osmoregulator, which increases a cell's ability to withstand extremes of temperature and desiccation. As an osmoprotectant, proline is used in many pharmaceutical and biotechnology applications. This is a Metacyc pathway for proline biosynthesis. And proline biosynthesis is fairly standard. Humans all the way to bacteria pretty much do the same thing, though eukaryotes do enter the pathway. The step prior to glutamate comes from different sources, but once it enters the pathway, it does the same for just about all organisms. The first two enzymes, gamma-glutamyl kinase and glutamate 5-semialdehyde dehydrogenase, form a multi-enzyme complex, and the first enzyme, which is encoded by the gene proB, it requires that interaction with the second enzyme to function, but the second enzyme, which is encoded by the gene proA, can work independently. Then we see that 5-glutamate or L-glutamate 5-semialdehyde forms 1-pyrroline 5-carboxylate. This is a spontaneous process. There's no enzyme for this reaction. And then 1-pyrroline 5-carboxylate is converted into proline, and that reaction is catalyzed by pyrroline 5-carboxylate reductase. And this enzyme is encoded by the gene proC. The gold coloration of these three enzymes indicates that functional evidence in Ecoli has been published for all three of these enzymes. Deutsch et al. In 1982 and 1984 cloned the proB, the proA, and proC genes into a special type of vector called an expression vector. An expression vector is one in which a suitable promoter has been positioned in front of the cloned insert and thereby allowing it to be transcribed and translated. So they are getting a protein product from the gene they cloned into something like a plasmid, I believe was the specific vector. And so they clone into this vector, and then they can study the expression of the clone. The clones were sequenced. The protein products of the genes were purified and also sequenced. The activity of the enzyme produced from each clone was measured under both inducible and inhibitory conditions. So consequently, functional evidence linking the nucleotide sequence of each gene and their respective enzyme activity have been made. So we're quite confident that the proA, proB, and proC genes produce the enzymes that we just described on a previous slide. I thought this was an interesting sideline to our proB, proA story. This figure, figure 2, is a schematic of the bacterial glutamate kinase, which is proB, and glutamyl phosphate reductase, which is the proA protein, as compared to a human version, which is called pyrroline 5-carboxylate synthase, or P5CS. In the top line, we see the Ecoli version of glutamate kinase and glutamyl phosphate reductase, two different proteins, one 367 amino acids long and the other 41 amino acids long. Oh, I don't think that can be true. I think it's 400 and something. I think I cut off a number there. And then just below it is the human P5CS protein. In eukaryotes, the proB and proA genes have fused, producing a single multi-domain protein of 795 amino acids. And as we see here, bacteria have two separate genes. But as noted earlier, glutamate kinase, or proB, is dependent on glutamyl phosphate reductase to function, where proamyl phosphate reductase, or proA, can function independently. What you see in this image is a schematic of the proB, proA operon found in E. coli. As you can see labeled on the top, there is a single promoter ahead of the proB gene. There is a single terminator behind the proA gene. It's anticipated that there is a Schindel-Garno sequence in front of the proB. However, I was not able to identify a Schindel-Garno sequence in front of proB using the GENI-ACT 6-frame viewer program. I'm sure there's one there, but there's not an identifiable one. But there is an identifiable Schindel-Garno sequence in front of the proA gene, which I think might have some implications for our future complementation assay, and I'll explain that in just a minute. Figure 4 represents or shows features of the proBA operon in Mrub. And if you look carefully, you'll notice that the numbers at the top are backwards. And that's because this region in Mrub is on the reverse polynucleotide strand, or also called the negative strand. To make the comparison to E. coli easier, I flipped the Mrub image so that now proB is on the left, and showing you that it would be transcribed from left to right, as opposed to in its normal state it would have been transcribed from right to left. So I hope that makes sense. As with E. coli, Mrub, proB, and proA are believed to be an operon. And because this pattern is maintained in Mrub's close relatives as well as most organisms in the bacterial domain. The green lines identify the start codon, and we can find those using the 6-frame viewer program. The red line indicates the stop codon, for proB. There is a putative Schindel-Garno in front of the proA, but it is much too far away from the proA start codon to be functional. As you probably remember, you need to be about 8 to 13 base pairs. The Schindel-Garno sequence needs to be 8 to 13 base pairs from its start codon to function. And so the blue region that we see in front of proA is actually within the proB gene. So unlikely to be a true Schindel-Garno. So since the Schindel-Garno sequence is not predicted within the standard region from our proA in Mrub, I'm a little concerned as to the impact of expressing our proA gene in our proBA clone for Mrub. E. coli, putting this clone into E. coli might only be able to express the proA gene regardless of the source if it has that Schindel-Garno sequence. So we have a little bit of problem going into our complementation work, and we'll just have to see how that works out. All right, this is Table 1. It is a bioinformatics comparison of the E. coli proA gene to Mrub 1079, which we are predicting is the comparable gene to E. coli proA. And notice that in this table I've only provided the E values for the Mrub, the Meiothermus ruber analyses, primarily just because of lack of space in my table. Now both E. coli and Meiothermus ruber are predicted to be localized to the cytoplasm. We tested this through Module 3. Signal P, TMHMM, Lipo-P, all those things clearly said cytoplasm for both. And we'd expect that because proline in other organisms is localized, proline production is localized to the cytoplasm. A blast alignment between E. coli proA and Mrub 1079 resulted in very low E value, which indicates strong sequence similarity, and as we know, sequence similarity would be highly indicative of functional similarity. Mrub 1079 is predicted to code for a protein and proline biosynthesis pathway as seen using the KEGG bioinformatics program. Both proteins have strong sequence similarity to the same protein families as determined by PFAM and TigerFAM. Now you might wonder about the PFAM PF00171 designation. This is a family of proteins that are dehydrogenases that act on aldehyde substrates, and members of this family use NADP as a cofactor. All of these apply to the glutamyl phosphate reductase enzyme. So even though it looks like a different name, it all fits with both E. coli and Mrub 1079. It all makes sense if you understand the reaction mechanism. Now, both proteins contain the same protein domain as determined by KEGG. They are predicted to catalyze the same enzyme reaction as indicated by the EC numbers. I believe this was acquired through Aspasia. Now, this enzyme has two common names, glutamyl phosphate reductase, or also glutamate 5-semialdehyde reductase. So we're not surprised to see these two names coming in here, depending on the bioinformatics program. And finally, Mrub 1079 produces a protein that shows strong sequence homology to several pro-A proteins in the PDB database, one of those being E. coli. So all this evidence, there is no contradictory evidence that would indicate that Mrub 1079 is anything other than the counterpart to E. coli pro-A. Table 2 is very similar to Table 1 in that it's a comparison of E. coli, but this time to Mrub 1080, which I'm predicting is both our proB genes, the reaction being from glutamate to glutamyl phosphate. And this is the enzyme that is produced from proB is glutamate kinase. As both E. coli and Mrub 1080 appear to be localized to the cytoplasm, again, exactly where you'd expect it to be. A blast comparison between the two gave us really a relatively high bit score, but most importantly, a very low E value, indicating sequence similarity. They both are predicted to be in the same biochemical pathway, that being through KEGG analysis. Now, the PFAM, we did get two PFAM domain categories coming up. One is an amino acid kinase, which is exactly what glutamate kinase does. It adds a phosphate group to glutamate to get glutamyl phosphate. There we go. Now, the other PFAM domain, 01472 PUA, again, a relatively low E value. This has something to do probably with the regulation of the proB gene. As we talked about earlier, proline biosynthesis is involved in osmoregulation, in the detoxification of oxygen-free radicals. So it's believed that this might be, that this enzyme might actually be involved in regulating other genes that might need proline, basically. The CDD, we get the same KEGG category, so glutamate 5 kinase, exactly what we'd expect. TigerFam, get the same protein family. EC number the same. PDB, there are actually proteins, including E. coli, that have been crystallized, and we pull those out of the PDB database. So again, everything would support that Mrub 1080 is in fact proB. Table 3 is a comparison of E. coli proC to Mrub 1345, and as with the previous two tables, there doesn't appear to be any contradictory evidence that Mrub 1345 isn't proC. Cytoplasmic localization, comparison between E. coli and Mrub gives a fairly low E value, which is what we'd expect. KEGG pathway predicts the same, it's involved in the same pathway. Same, all these KEGG numbers, TigerFam, EC numbers all point to the same enzyme, pyrroline 5 carboxylate reductase. It should be noted that PFAM pulled out an oxidoreductase, NADP oxidoreductase coenzyme. Is exactly what pyrroline 5 carboxylate reductase does. So again, nothing inconsistent with the function of Mrub 1345. Figure 5 is a phylogenetic tree of Mrub, I guess it would be Mrub 1345 or what we are proposing is proC. I moved this figure ahead of proA and proB because this one is a great representation of no horizontal gene transfers. It's everything, nearly the entire Deinococcus thermus phylum, at least those genomes that have been sequenced, are represented in this tree, and you have to go really far back in the tree to get an organism outside this phylum. So all the organisms in this tree point to the same phyla. Mrub 1, 3, 4, 5 looks like no horizontal gene transfer. Figure 6 is a phylogenetic tree of Mrub 1079, which we're predicting is proA. Mrub 1079 appears most closely related to other Meiothermus and Thermus species, though it should be noted that some species in this phyla are not on this phylogenetic tree, so that's kind of curious. Because Mrub is around other Meiothermus and other Thermus, there's no indication of at least recent horizontal gene transfer. However, there is a hint of HGT in the distant past. If you look at the top third of this phylogenetic tree, we see several species from another phylum called Proteobacteria. So it would be interesting to see how the flanking genes in this region of Mrub, as well as its other organisms on this phylogenetic tree, how they compare to each other. Do they look as though all these organisms have the same basic series of genes? Because, as we talked about earlier, HGT normally involves the transfer of a region of DNA, not just one gene or a piece of a gene. So between that and possibly looking at percent GC content, so that's going to be a little problematic. As you know, Mrub has a higher percent GC content just because it's a thermophilic organism, and that's an adaptation to living in a warmer environment. So if HGT has occurred in the distant past, chances are this gene has already adapted to the more warmer environment, and so the percent GC content is probably not going to be particularly helpful in studying HGT. As with Mrub 1079, or the putative proA gene from figure 6, Mrub 1080, or what we're proposing is proB, is most closely related to other Deinococcus thermus genera, but again, there is a hint of HGT in the distant past. At the top of the tree are several species within the Proteobacteria phyla, and just as before, it would be interesting to see how the gene neighborhoods compare for these distantly related species as compared to Mrub. This ends the discussion of the gene annotation component of proline biosynthesis. If you're interested in learning how the molecular genetics class looked at the functional genomics aspect of proline biosynthesis, then please listen to the next video in this series.